sometimes are, or agar. Agar is a jelly-like substance, obtained from red algae. 1. Agar is a mixture of two components, the linear polysaccharide agarose, and a heterogeneous mixture of smaller molecules called agaropectin. 2. It forms the supporting structure in the cell walls of certain species of algae, and is released on boiling. These algae are known as agarophytes, and belong to the Rhodophyta, red algae, phylum. 3. 4. Agar has been used as an ingredient in desserts throughout Asia, and also as a solid substrate to contain culture media for microbiological work. Agar can be used as a laxative, an appetite suppressant, a vegetarian substitute for gelatin, a thickener for soups, in fruit preserves, ice cream, and other desserts, as a clarifying agent in brewing, and for sizing paper and fabrics. 5. The gelling agent in agar is an unbranched polysaccharide obtained from the cell walls of some species of red algae, primarily from Tungusa, Gelatiaceae, and Aganori, Gracilaria. For commercial purposes, it is derived primarily from Aganori. 6. In chemical terms, agar is a polymer made up of subunits of the sugar galactose. Contents History Edit. The structure of an agarose polymer. Agar may have been discovered in Japan in 1658 by Minotaur,
味濃太郎作為文
an innkeeper in current Fushimi-ku, Kyoto, who, according to legend, was said to have discarded surplus seaweed soup and noticed that it gelled later after a winter night's freezing. 7. Over the following centuries, agar became a common gelling agent in several Southeast Asian cuisines. 8. Agar was first subjected to chemical analysis in 1859 by the French chemist Anselme Paton, who had obtained agar from the marine algae Gillidium corneum. 9. Beginning in the late 19th century, agar began to be used heavily as a solid medium for growing various microbes. Agar was first described for use in microbiology in 1882 by the German microbiologist Walter Hesse, an assistant working in Robert Koch's laboratory, on the suggestion of his wife Fanny Hesse. 10. 11. Agar quickly supplanted gelatin as the base of microbiological media, due to its higher melting temperature, allowing microbes to be grown at higher temperatures without the media liquefying. 12. With its newfound use in microbiology, agar production quickly increased. This production centered on Japan, which produced most of the world's agar until World War II. 13. However, with the outbreak of World War II, many nations were forced to establish domestic agar industries in order to continue microbiological research. 13. Around the time of World War II, approximately 2,500 tons of agar were produced annually. 13. By the mid-1970s, production worldwide had increased dramatically to approximately 10,000 tons each year. 13. Since then, production of agar has fluctuated due to unstable and sometimes overutilized seaweed populations. 14. Etymology Edit. The word agar comes from agar, the Malay name for red algae, Yagardina, Gracilaria, from which the jelly is produced. 15. It is also known as Canton, Japanese,
寒天
from the phrase Kanzarashita Korten,
寒水心態
or cold exposed agar, Japanese isinglass, Ceylon moss or Jaffna moss. 16. Gracilaria lichenoides is specifically referred to as agal-agal or Ceylon agar. 17. Composition Edit. Agar consists of a mixture of two polysaccharides, agarose and agaropectin, with agarose making up about 70% of the mixture. 18. Agarose is a linear polymer, made up of repeating units of agarobiose, a disaccharide made up of D-galactose and 3, 6, anhydroL-galactopyranose. 19. Agaropectin is a heterogeneous mixture of smaller molecules that occur in lesser amounts, and is made up of alternating units of D-galactose and L-galactose heavily modified with acidic side groups, such as sulfate and pyruvate. 20. 18. 19. Agar exhibits hysteresis, melting at 85 degrees Celsius, 358 K, 185 degrees Fahrenheit, and solidifying from 32-40 degrees Celsius, 305-313 K, 90-104 degrees Fahrenheit. 21. This property lends a suitable balance between easy melting and good gel stability at relatively high temperatures. Since many scientific applications require incubation at temperatures close to human body temperature, 37 degrees Celsius, agar is more appropriate than other solidifying agents that melt at this temperature, such as gelatin. Uses edit. Culinary edit. Sago at gulaman in Filipino cuisine is made from agar, gulaman, pearl sago, and fruit juice flavored with pandan agar. Agar is a natural vegetable gelatin counterpart. White and semi-translucent, it is sold in packages as washed and dried strips or in powdered form. It can be used to make jellies, puddings, and custards. For making jelly, it is boiled in water until the solids dissolve. Sweetener, flavoring, coloring, fruit or vegetables are then added and the liquid is poured into molds to be served as desserts and vegetable aspects, or incorporated with other desserts, such as a jelly layer in a cake. Agar is approximately 80% fiber, so it can serve as an intestinal regulator. Its bulking quality has been behind fad diets in Asia, for example the Canton, the Japanese word for agar for diet. Once ingested, Canton triples in size and absorbs water. This results in the consumers feeling fuller. This diet has recently received some press coverage in the United States as well. The diet has shown promise in obesity studies. 22. One use of agar in Japanese cuisine, wagashi, is enmitsu, a dessert made of small cubes of agar jelly and served in a bowl with various fruits or other ingredients. It is also the main ingredient in mizuyakin, another popular Japanese food. In Philippine cuisine, it is used to make the jelly bars in the various gulaman refreshments or desserts such as sago gulaman, buko pandan, agar flan, halo, and the black and red gulaman used in various fruit salads. In Vietnamese cuisine, jellies made of flavored layers of agar, called thch, are a popular dessert, and are often made in ornate molds for special occasions. In Indian cuisine, agar is known as China grass and is used for making desserts. In Burmese cuisine, a sweet jelly known as kyak cha, tat, is made from agar. In Russia, it is used in addition or as a replacement to pectin in jams and marmalades, as a substitute to gelatin for its superior gelling properties, and as a strengthening ingredient in soufflés and custards. Another use of agar is in tichay malako, bird's milk, a rich jellified custard, or soft meringue, used as a cake filling or chocolate glazed as individual sweets. Agar may also be used as the gelling agent in gel clarification, a culinary technique used to clarify stocks, sauces, and other liquids. Mexico has traditional candies made out of agar gelatin, most of them in colorful, half-circle shapes that resemble a melon or watermelon fruit slice, and commonly covered with sugar. They are known in Spanish as dulce de agar sweets. Agar is an allowed non-organic, non-synthetic additive used as a thickener, gelling agent, texturizer, moisturizer, emulsifier, flavor enhancer, and absorbent in certified organic foods. 23. Microbiology edit. 100 mm diameter petri dishes containing agar gel for bacterial culture. Main article. Agar plate. An agar plate or petri dish is used to provide a growth medium using a mix of agar and other nutrients in which microorganisms, including bacteria and fungi, can be cultured and observed under the microscope. Agar is indigestible for many organisms so that microbial growth does not affect the gel used and it remains stable. Agar is typically sold commercially as a powder that can be mixed with water and prepared similarly to gelatin before use as a growth medium. Other ingredients are added to the agar to meet the nutritional needs of the microbes. Many specific formulations are available because some microbes prefer certain environmental conditions over others. Agar is often dispensed using a sterile media dispenser. Motility assays edit. As a gel, an agar or agarose medium is porous and therefore can be used to measure microorganism motility and mobility. The gel's porosity is directly related to the concentration of agarose in the medium, so various levels of effective viscosity, from the cell's point of view, can be selected, depending on the experimental objectives. A common identification assay involves culturing a sample of the organism deep within a block of nutrient agar. Cells will attempt to grow within the gel structure. Motile species will be able to migrate, albeit slowly, throughout the gel, and infiltration rates can then be visualized, whereas non-modal species will show growth only along the not-empty path introduced by the invasive initial sample deposition. Another setup commonly used for measuring chemotaxis and chemokinesis utilizes the under-agarose cell migration assay, whereby a layer of agarose gel is placed between a cell population and a chemoattractant. As a concentration gradient develops from the diffusion of the chemoattractant into the gel, various cell populations requiring different stimulation levels to migrate can then be visualized over time using microphotography as they tunnel upward through the gel against gravity along the gradient. Plant biology edit. Fiscometrella patens plants growing azenically in vitro on agar plates, petri dish, 9 cm diameter. Research-grade agar is used extensively in plant biology as it is supplemented with a nutrient and vitamin mixture that allows for seedling germination in petri dishes under sterile conditions, given that the seeds are sterilized as well. Nutrient and vitamin supplementation for Arabidopsis thaliana is standard across most experimental conditions. Neuroscheidenskog, Mies, nutrient mix and Gamborg's B5 vitamin mix in general are used. A 1.0% agar/0.44% Mies plus vitamin D H2O solution is suitable for growth media between normal growth temps. When using agar, within any growth medium, it is important to know that the solidification of the agar is pH dependent. The optimal range for solidification is between 5.4 to 5.7, 24. Usually, the application of CO is needed to increase the pH to this range. A general guideline is about 600 microl 0.1 m CO per 250 milliliters grams. This entire mixture can be sterilized using the liquid cycle of an autoclave. This medium nicely lends itself to the application of specific concentrations of phytohormones etc. to induce specific growth patterns, and that one can easily prepare a solution containing the desired amount of hormone. Add it to the known volume of GM, and autoclave to both sterilize and evaporate off any solvent that may have been used to dissolve the often polar hormones. This hormone GM solution can be spread across the surface of petri dishes sown with germinated and/or etiolated seedlings. Experiments with the moss Fiscometrella patens, however, have shown that choice of the gelling agent, agar or gelrite, does influence phytohormone sensitivity of the plant cell culture. 25. Other uses. Added agar is used as an impression material in dentistry, as a medium to precisely orient the tissue specimen and secure it by agar pre-embedding, especially useful for small endoscopy biopsy specimens, for histopathology processing. 26. To make salt bridges and gel plugs for use in electrochemistry. In formicariums as a transparent substitute for sand and a source of nutrition. As a natural ingredient to form modeling clay for young children to play with. As an allowed biofertilizer component in organic farming. 27. As a substrate for precipitin reactions in immunology. Gelidium agar is used primarily for bacteriological plates. Gracilaria agar is used mainly in food applications. In 2016, Amam, a Japanese company, developed a prototype for agar-based commercial packaging system called Agar Plasticity, intended as a replacement for oil-based plastic packaging.